Okay. So hello everybody. It's my pleasure to start this meeting and a little bit history of it. So it all started, I think, 11 years ago when after we received ONR Muri grant in 2013, I believe, we had the first kickoff meeting. And ONR program manager, Tom McKenna, who unfortunately couldn't make it in the last moment to this meeting, asked us to organize sort of a kickoff meeting. And we decided instead of just having students and us talking, we tried to make it like some big event, at least first day. And the second day was like more regular one. So anyway, this is how it started. And then we continued doing it every year. I think we skipped 1 year during COVID, and 1 year it was online, kind of virtual. But I counted, this is the 10th time we organized this meeting, so it's kind of anniversary. So it's so again, thank you so much everybody for coming, and we have great list of speakers, and you have schedules and so on. We have a little change, kind of we switch first two speakers because first speaker a bit late, but nothing major, no other major changes. So with that, I would like to invite our first speaker, Eric Halgren, who is my long-term collaborator, and it's a real pleasure to introduce him. He has been doing lots of fantastic work, working with human data, and I think the last set of work about ripples is one thing which is I'm not sure if you're talking about ripples or not, but for me it's kind of a topic which remains a mystery. I mean, usually you say, okay, I understand how it works, at least conceptually, we can model it or something. So this type of sort of results we're getting for the last few years, I have no idea how to explain. And again, maybe he's talking about something more exciting, but so Eric, please. Yeah.
Thank you. Thank you, everyone, and thank the audience for not moaning when you found out that your first speaker was not going to be Yuri Buzhaki. But I'm very honored to be here, and thank you, Terry and Maxime. I can't imagine anything that would be more thrilling than something you can't explain. So that is what I'm going to try to do. Because, in fact, I am going to talk about ripples, or co-ripples, which they're 90 hertz, 100 millisecond LFP oscillations in humans. And the mechanism, I'm going to argue, is coupled oscillators. We have some new data about that. And the purpose is cortical integration, or, more broadly, how to get rid of the goddamn homunculus. And so, warning, you're about to view unpublished data and wild hypotheses regarding emergent phenomena. If that makes you uncomfortable, please hide your heads under the desk. So I chose to talk about this because understanding, making rigorous emergent phenomena is what I hope the interaction between computational and empirical neuroscientists will lead to. Okay, so as Maxime said, we have been, let's see if this works. No, but I can, huh? Do the pointer. Yeah, the pointer's better. We started looking at ripples over 5 years ago. And first, we just tried to replicate what Yuri had found. Other people had looked at in humans, but basically we could find these ripples during non-REM sleep, during sharp wave ripples during sharp waves. And we found that similar phenomena were occurring in the cortex during non-REM sleep on the upslope of upstates, and were occurring also during waking. And they were happening all over the cortex. And in subsequent work, we found that we can record them with depth electrodes, surface electrodes, microelectrode arrays, Utah arrays, laminar arrays, surface microgrids, fine wires, neuropixels. And we find them in epileptic patients, cancer patients, paralyzed patients, a variety of people who get electrodes inside their head for clinical purposes, but we are lucky to get the opportunity to record from them. So I think, in my mind, they're very well established. Recently, we published a analysis of how tasks modulate them. So the basic comparison here is between the response to words and target words, which are animals. Person presses a key. They're coming really fast, and these are just like 5% of the trials. So the plots I'm going to be showing you are lines, and these lines indicate a significant increase during this period of time to this condition. And if there's no line, there's no significant increase over baseline in this case. And the color of the line is the ratio to the baseline, which for this, for example, is 10-fold increase from baseline. That's somewhat higher than you see with fMRI. And and the thickness of the line is the density of the co-ripples. So basically, if you look at all of the words, or in this case, all the non-target novel words, you see this connection between the fusiform gyrus, which is the visual word form area, and on the left side, this will all be on the left side right now, and all of these areas, basically all of the areas where we're recording from. We divided up for this purpose the brain into these areas. And, but later it just totally peters out. However, if you look at the target compared to non-target novel words, well, there's not much here because targets don't evoke much differentially. But when you get out to here, just before the response, it's like everybody's talking to everybody at very high rates and very significantly. I would also point out that there's no connection between these areas except with the fusiform. So we call this seeding, and we call this integrating. Seeding, and here we have the time course in milliseconds from the fusiform to everywhere else, and between each of these areas and everywhere else. And you see, basically, it's the fusiform that's doing it to the non-target novel words and projecting all over the place. And then later, to the target novel words, many areas are getting in the picture. So we looked at the phase lag between the structures and their phase locking. So phase locking means, that between trials, the oscillations are on top of each other. If you have phase lag, if you have phase locking at zero lag. And so the lines indicate that there is significant phase locking value. So that's basically between everywhere. And the fact that the thickness is how significant they are. And then the color of the lines is the phase lag, and you see it's green almost always because that's zero. So there is strong phase locking at zero phase lag. And we've run 1 million controls, and this particular control was doing bipolar instead of extracranial reference, and you get the same results. We also looked at the effects of being correct. Hi, welcome. Yeah, I'm the poor imitation, but we'll get Yuri soon. So this is the comparison between, this is a reading task. And this is a comparison between the correct and incorrect trials. So in this case, we're comparing between true positives where you press to frog and false positives where you press to some other word, non-animal words. And you see, going back from the key press, that correct trials evoke much stronger co-rippling between parietal and prefrontal and Rolandic areas. And this is the time course of that. And this is also seen between true positives and false negatives. Okay, so that's recently published. Now, this is unpublished data where we have looked at open source data that is bilateral unit LFP recordings during the Sternberg paradigm. These are recorded with what are called Benke-Fried probes, which are depth electrodes which go down from the skull to, in a tube, to these areas near the midline, and there are 5 sites selected on each side. So there are 10 Benke-Fried probes in each subject bilaterally, okay? And this is about 1,500 single units. In this case, during the Sternberg paradigm, which 3 images are presented, there's a delay period, and then an image is presented and the person needs to respond if that was one of those 3. Sometimes it's just 1 image, sometimes it's 3. So here are the co-ripples between these areas. In this case, it's between left and right ACC. So the areas are orbitofrontal, anterior cingulate, pre-SMA, pre-supplementary motor, amygdala, and hippocampus. And here are all the links, and they basically are all going up, and they're all differing between baseline and in all the different conditions. And here I'm just showing the relationship to memory load. And the co-ripples, the dots in this significance matrix are generally increasing during 3 stimulus versus 1 stimulus trials. And that's not seen very much in higher or lower gamma. So then we wanted to see how that was related to single unit activity. And the first thing we wanted to find out was whether co-firing is affected between these distant areas by co-ripples. And so we plotted the distance between the units and the co-firing z-score. And you can see that during co-ripples, it's quite a bit higher than during no ripples, and that is consistent across these long distances. So, I mean, we're talking about like from, hither to yon, right? These are long distances, over 200 millimeters by axon length, and yet the co-ripples are correlated with a big increase in co-firing. So we wanted to look more carefully then at the co-firing, and what you see here is the baseline period, and this is the rate in the baseline period for the different pairs of sites. And this is, again, the co-firing, which in this case is not really changing in the different task conditions. But then we divided this co-firing, into when it was happening during co-ripples versus when it wasn't. And there's a big increase during the co-ripples, and sometimes a bit of a decrease relative to baseline during the no ripples. And again, about 1,500 units. So the observation was that task-modulated increased co-firing only occurs during co-ripples. And our assumption is that task-relevant cortical integration requires task-modulated co-firing. That seems obvious to me, but I don't really see that ever expressed. And so our conclusion is that co-ripples may organize task-relevant cortical integration. We also looked to see whether integration was enhanced by co-ripples by asking whether co-ripples increase cortical-cortical integration as measured by co-prediction of firing during free behavior. So this is spontaneous activity. Here we're using Utah rays, which have 96 of these, 2 millimeter long sharpened probes going into layer 3, more or less. And this is a single subject with tetraplegia, separated with 2 Utah rays separated by about 12 millimeters. And then we have 2 probes here in 2 different subjects with epilepsy. And we cut the units. Here's an example of recording from 2 different sites. And we asked whether the history of firing by B would predict whether A would have a spike. So here is that prediction in location E1 when you have co-ripples, and here is the prediction when you have no ripples. This is the distance within that array. And the results are consistent. In these, you have the 2 arrays, and it is maintained at 12 millimeters. So here's another set of studies we have where we are, again, have the Benke-Fried probes, open access. This is from Uli Rudeshire's lab, very generous sharing of data. And in this case, the person watches a movie and makes a judgment whether it's new or old and gives their certainty. And there's an increase in confident versus unsure co-rippling. And I'll talk more about the units later, but I wanted to bring this up because I'm just trying to go through the different tasks we have with co-ripples. And I wanted to introduce one more task, which is simply listening and making simple judgments on consonant vowel syllables. In the posterior superior temporal gyrus, here we have a electrode array with 1,024 channels with a 200 micron pitch. We also have 50 micron pitch, smaller arrays. And I just want to point out one aspect of that recordings. Using a non-negative matrix factorization, we find these modules. If you look at the activity during these modules, this is the amplitude in the ripple band, and this is the activity of a row of channels, this row right here that goes between 2 modules, modules 2 and 7, and this is time. You can see that the time of the response, not just what it responds to, is very different, which is kind of amazing. Here you're right next to each other, and they're responding at very different latencies. And here is the PLV showing how it's focal and identifies these 2 modules. So average diameter is 1.7 millimeters. If the entire cortical surface were tiled with them, there'd be about 128,000, each with around 96,000 neurons. And that's, completely unknown whether it extends beyond this particular type of special cortex, but we're going to assume that for the rest of the discussion. All right, so here's a summary of the task correlates of co-ripples that we have found so far. And I'm not going to go into it in detail, but just say it seems to be correlated with making a difficult decision correctly. We can talk about why later if you want. But that's not terribly important. Here's our interim summary of the characteristics of cortical co-ripples in humans. They occur throughout the human cortex in both sleep and waking. They're recorded with a wide variety of macro and micro electrodes. They're recorded in people with different conditions, and are organized perhaps in 1.7 millimeter diameter modules. And their possible integrative powers are zero phase locking, zero lag phase locking, strong modulation in density, location, and timing by tasks, strong association with correct task performance, and we think they appear to enable distant cortical locations to have task-related co-firing and enable neurons in different cortical locations to predict each other's firing. So, how does this come about? There are two obvious hypotheses. One is that it's some sort of emergent property in the cortex, and the other is that it's driven by the thalamus. You know, and the thalamus driving would be there'd be synchronous pulses coming out at 90 hertz and driving these different cortical cells. All right, we recently published a paper in PLOS Biology that looks at the thalamus and cortex during co-ripples, and we found that the thalamus indeed does ripple. It's not as often as the cortex, but it does. This was in non-REM. They are related in the thalamus to spindles and to upstates, which like in the cortex. And if you look at the thalamocortical co-occurrence or phase locking of spindles and upstates, that has an influence on how much cortical co-rippling is occurring. But if you look for phase locking between the thalamus and the cortex, it's rare or doesn't occur at all. So we don't have evidence for this. We have evidence for this, that the thalamus modulates the co-rippling, but doesn't actually drive it on a wave-by-wave basis. So let's see if we can get any more indirect evidence. Here's one dive into the literature. If you add up all of the subcortical structures interacting with the neocortex, some of which are listed here, it's about 5% of the sources of interaction that would occur by looking within the neocortex. So this is in humans. It's going to be very different in a mouse. But in a human, I think it's pretty clear that the vast majority of connections are cortical. So if you actually take the number of projection cells in the thalamus and compare it to the number of neocortical cells, you get about 720 cortical cells for every thalamic projection cell. So the implication is that anything the thalamus does to the cortex is probably going to be low resolution. And it's going to require more white matter because you have to control when it happens, so you need two connections. And we're all out of white matter. we really are white matter limited. And it requires longer transmission time, obviously, due to the length and the synaptic delay. So that's, why it would be unlikely. Well, on the other hand, is it likely that cortical would be? Well, long-range connections are very rare in the cortex. They drop off exponentially. And if you look at just the numbers, they're also very low at a distance. The probability of monosynaptic pathway between two specific neurons is really near zero, because you have billions of them, and just you have thousands of synapses coming out from any given neuron. So it's close to zero, but the number of polysynaptic pathways is quasi-infinite. So that's just the nature of cortical-cortical connectivity. Pyramidal cells only receive around 5 to 20% of their excitatory input locally, very locally. But it seems that most of the pyramidal connectivity may be between layer 3B cells in rosettes at 1 or 0.5 to 10 millimeters. That's my reading of a lot of literature, and it's unknown, and I think that's a critical thing to find out. But anyway, the hypothesis would be the synchronization relies on a net of mid-range L3B connections. Fortunately, multipath bidirectional short-range connectivity is probably sufficient. 3.5 centuries ago, Huygens hung a couple pendulums from a rigid beam and found that if he sets them swinging, that they would synchronize. And 3.5 centuries later, Bazhanov did a large cortical model which found the same thing, and showed that requires just this two-dimensional connectivity between the elements. So fortunately, we have stumbled upon what I think is a more direct demonstration that the cortical-cortical synchrony is happening instead of the thalamocortical driving. And this is explained here. If you had a synchrony from a central pacemaker, thalamic pacemaker, it's projecting here to 3 different cortical areas. And in each area, they're firing here at the peak of the spike. We call this B and this an A. And then if you do a cross-correlogram of the spikes in A relative to spikes in B, you will see then peaks at every peak of the shared zero lag ripple, right? I hope you're all acquainted with cross-correlograms. So on the other hand, if you have a network with delays, because it's cortical-cortical, then it's going to take time to get from a spike in A to a spike in B, and again, time to get from a spike in B to a spike in A, and that time is between the peaks of the ripple oscillations. So if you do a cross-correlogram, you're going to get a dip here at t equals 0, and these peaks at t equals -1 and t equals +1. Is that clear? Okay. Well, what do you see? These are the two hypotheses, and what you see is this dip. This is the cross-correlogram between bundles in the movie and in the Sternberg tasks. This is Ilya's work. And so there's a dip. And I think that's pretty strong evidence that it's not being broadcast from a single point. So how might co-ripples emerge as from cortical interactions? The biggest modulation is something that the binding bisynchrony people have talked about for a very long time, which is this. Shown here that there's an increased probability of firing at all peaks if you look locally at a cell to its local oscillation. And that's true for both pyramidal and interneurons, and they have this characteristic delay between them. which urea is shown in mice. And I'm not going to go too much into this, but there's also coincidence detection where cells arriving have their action potentials arriving at a third place, and this is something that Terry showed very well, is that this can be a very strong effect on whether area 3 will fire, is if there are synchronous EPSPs arriving together. And so how do you get the other 16 billion cortical neurons to listen to you? I think that's a really fundamental problem of neuroscience. Basically, I think singing in a group on pitch and on the beat is the answer, and we'll delve into that more. And then, LFPs oscillate, spikes couple them. I think it's obvious how this is a coupled oscillator, and I won't need to go into that. All right. Exponential growth is typical of some coupled oscillators. And here I'm just going to talk about one example that's shown down here. This, as you remember, is the response to the target novel words. Here is the time course between the prefrontal and parietal areas in the density of the co-ripples. And it starts low and goes high, and just look how it goes up. So if you take from 400 to 640 and fit a curve to that, it is pretty close to a exponential with a doubling time of about 60 milliseconds, which would require that the ripples increase about, for every ripple cycle, that the number of co-ripples increases about 14%. Okay? So we'll return to that number in a little while. All right, so here's, we can add a few things on to the interim summary. The mechanism we think is cortical-cortical cross-speak communication of coupled oscillators connected with multiple medium-length axons, phase modulation coincidence detection, and basically this is coupled oscillators leading to resonance, and exponential growth in co-rippling prior to making a decision. Now, I'm going to talk in my, I guess I have a few more minutes, about hierarchical predictive processing, attention, lower frequency waves, and traveling waves, which are all alternative mechanisms for cortical-cortical integration. And I think we all know Fellman and Van Essen's diagram of the macaque visual system, noting its hierarchical organization, and Markov's analysis of its connectivity, and the predictive coding of a feed-forward and feedback system. The problem is that is illustrated with this. This is myelination of the cortex, and myelination is a marker of hierarchical level. And you can see here in the visual system of the macaque, it is a variation in the myelination, okay? The human, it's much, much smaller, and these hierarchically organized areas are just a tiny bit of the human brain. Most of the brain is not hierarchical. So having a integration system based on hierarchy is not going to work. This is an elaboration of that system which uses attention. And I think the homunculus raises its head again. Where does attention come from, and how does increased firing in visual areas result in particular behavioral choices? I won't go into that, but Ralsma has a nice review of that. So I want to consider the task again. We have some I/O module where it's reasonable to suppose that there's a focal sequential hierarchical convergent of labeled lines, activation of labeled lines. Like, the V1 has cells that respond to these stimuli. You combine them, and they will make these letters. You combine the letters, they'll make a word form. You can imagine that all happens like dung, 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 right? But beyond that, what about meaning, the instructions, the action? And that, we know, is, about over 3/4 of the time from the stimulus presentation to the response. And we know that that is associated with widespread evoked potentials in the association areas, which is most of the brain. So the idea is that the hierarchical processing, which is happening here before the co-ripples start changing, is there, but that what's happening after that is co-ripples. Now, we looked for evidence for that by trying to measure co-high gamma, which was even higher frequency than co-ripples and was asynchronous, and their co-occurrence. And we found that, yes, they occur very early, but after that, they don't occur. And they are much less modulated by the test than co-ripples. But it seems that even in the fusiform gyrus, they occur before the ripples and may, in fact, trigger them through depolarization, as Yuri has shown happens in the cortex. So that's our hypothesis, that's sequential. All right, the alert viewers might have noted earlier a problem in the dip I was showing. So here's the dips we were looking at, right here and here, in the cross-correlogram. And here is the idea of how that would happen is these two bumps are 2 pi away, but these lines are at the 2 pi marks, and they're not on those lines. In fact, they're only about 1.2 pi distant from 0. So why? Well, one possibility would be that the sending spikes, the spikes in A that tend to trigger something in B, are coming mainly from the latter part of the ripple wave, and the ones that get triggered in B tend to occur more often before the peak of B's oscillation. All right, so that would result in this less than 2 pi, which is between the peaks. So we have some very preliminary data looking at the phase of A with respect to, of such spikes in A, with respect to its peak, and B with respect to its peak, and they are consistent with that interpretation. But, we have a lot more to do. So the idea is here. An early spike in B is triggered by a late spike in A at the conclusion of the processing in the prior cycle, and that this late spike in B at the conclusion of its prior cycle, of its processing cycle, goes on to some other place. Now, how do we know that processing occurs within in the location? So you can look at, by processing, we can look for co-firing, and these are within the bundle, and you get the peak at 0. And these are co-spikes between cells that are different cells. It's not the same cell, it's different cells. 5 minutes, got it. Thank you. And just showing this is also dependent on co-ripples because it's the no ripple level. All right, So we have intermodule processing, which is this conclusion of the previous cycle, going from many modules converging on other modules, and within module, intermodule processing. And each one of these, like I say, is, around 3 to 10K modules, and there are about, large numbers of possible connections, and then that happens again. And these are the cortical consensus at different times. Then you have the conduction delay acts as a memory store where the consensus cortical pattern output from each ripple peak is feed forward as input to multiple future peaks, which I think is something like Terry's going to talk about in transformers, maybe. Just to conclude that segment, periodic suppression of the local circuit forces it to participate in a larger self-organizing, co-rippling network. Local networks are much stronger than distant, so recurrent local networks will dominate unless they're periodically suppressed, basically by basket cell inhibition. And the input local circuit at the upslope is thus predominantly from distant neurons because the local neurons are turned off in the 6 milliseconds prior, and the effective output from the local circuit at the ripple oscillation downslope is predominantly to distant neurons because the local neurons are not receptive in the 6 milliseconds after. So this provides the rhythm between local and distant processing. And we need to realize how hard human cortical pyramidal cells are to fire, and just how this input during co-ripples is a synchronized blast from other zero-phase lag locked modules, and that really is going to make it strong. And modules permit that because they organize it. Okay, so why would more cycles be better? Basically, we see this exponential rise in 3 different locations. And like I said before, it was a 14% ripple cycle. It would be about 800% at 4 hertz, so in one cycle, if you were going to explain this rise in excitation. And the idea is that is what's going to be useful. So why not traveling waves? I don't have much space, but I would say that it's because it gives a lot more freedom to associations, and it allows the exponential growth. All right, so the conclusion is zero lag, phase locked, cortical co-ripples in humans are real, ubiquitous, emergent, effective, and integrative. And here are the people who did the work. Thank you.
Yeah, so if you want a question, come up to this microphone because we want to record it. Okay, so I'll start with the first question, and people should line up if they have a question. Mark? No question, okay. So this is wonderful that we have this incredible, you've taken it, the precision of the cortex down to a few milliseconds, which, would have been heresy in the last century, right? The rate code theory of how the cortex works. So, the evidence you have is really quite compelling. Now, in terms of the mechanism for creating a ripple within a module, you alluded that it might have something to do with basket cells, right? And that's also been used to explain regular gamma, which is 30 to 80. So do you see, there are different basket cells, or is there another type of inhibitory cell involved? What's your view on that?
I think probably Yuri is a much better person to answer that because he wrote the review on it. But I think we don't know enough about all of the dynamics of human interneurons. It's something that is emerging, but I think they're both basket cells, and that's based on the data we have, the data everyone
see
has.
ripples in monkeys, right?
Yeah.
It's not exclusive. It's the same
No.
frequency band. You don't see ripples in monkeys.
You do, and you obviously you see them in mice, and they're at 160 hertz.
Okay, different species have different frequencies. Now 160 hertz is going way beyond the basket cell in terms of that as a mechanism.
Well, I think, I don't know, are you going to talk about this in your talk? No. But that is the mechanism also. It's part of the mechanism, and I think if anybody could respond how you're able to get that high a frequency out of it, it would be Yuri. I can't.
Well, so it could be intrinsic mechanisms within the neuron itself. In other words, ion channels have that kind of time.
It's all going to be a combination of intrinsic and
Right.
and network, and it's a combination of the pyramidal interneuron pyramidal loop, but it's even more the interneuron loop, where the co-firing of interneurons create a oscillation in the interneurons because they are densely interconnected, and that also leads to
Good. That's another
an oscillation. So
possible
it's
mechanism. Right.
yeah,
Right.
but they work together, I think, is what the
Right.
overall evidence is, although I'm not an
Yeah,
expert at that.
so it is possible for inhibitory cells to also become synchronized. That would be an interesting, to add that in, right, to the
Yeah,
model. Yeah,
absolutely.
great.
Yeah, really fascinating. I was wondering, you mentioned earlier about different oscillations and how there may be some kind of synchronization with different oscillations and ripples being kind of co-occurring with things like spindles and different events. Did you see that kind of as ubiquitously with it throughout the cortex? Did you see a kind of a common co-occurrence of these co-ripples with other events? Was it like a ubiquitous thing that you saw, same as the co-ripples?
It's very strong in non-REM sleep.
Right.
The relationship, and especially with the upslope just before you get to the peak of a upstate in the cortex. That's characteristically where ripples occur, and that's also where spindles occur. But we haven't found, we haven't looked enough for co-occurrence with slower waves in waking.
Right. Yeah. Yeah. I'd be curious to see if there's some kind of other, oscillation or something like that, which would.
Yeah.
link them together.
Yeah. I mean, there must be multi-scale nested interactions, but we know they're going on in these tasks. We just, if you look grossly, you don't see them, but we need to look more finely.
Thank you.
Hi, Eric. That was a great talk.
Hi, thank you.
I was wondering if you've had a chance to look at any effects of like interhemispheric communication in this at all. I know you had a couple plots where you were showing the two sides of the brain, but if you've seen any sort of systemic differences in leading and lagging in hemispheres or anything like that for the co-ripple communication?
We haven't, and we haven't looked at it carefully enough.
Okay.
Not all of the data is available in the bilateral implantations, but we're going to ask for it.
Okay, great.
Hello, thank you so much for a fascinating talk. So my question was, do we think that the benefit of these co-ripples that we see, one of the benefits might be evolutionarily to like learn, because you mentioned one point that co-ripples are strongly affected when we recall a memory, and when we recall, we are like learning something better. So do we see one of the purposes of this might be to remember better?
Well, you can see the amount of spikes that occur, and the fact that like I was showing the spikes occurring, the firing, because the cross-correlograms are, you know, firing relative to B of A. And you can see that there is a lot of spikes that are firing within a very short period of time. Even, you know, if you're 11 milliseconds out, you're still within the range of STDP. And in other cases, you are within, you know, a millisecond or two. We were very careful that we weren't recording the same spike with two different electrodes, but you have this co-firing at very small intervals, which, you know, could lead to STDP. So yes, this could be contributing to that.
Okay. The second question is, so this what we saw was in healthy human brain, right? And a lot of the work experiments to find some insight, we also look into patients who have something that we know is not right. So what can we tell about these co-ripple activity in a patient?
Yeah. So there are three types of patients we've seen this in. All of the Benke-Fried and half of the Utah array recordings and all of the SEEG and ECOG were in patients who had chronic epilepsy. And we were fastidious in trying to get rid of the contacts that might be epileptiform, have epileptiform activity, or if there was occasional epileptiform activity in a contact, to get rid of those epics. But it always is a concern. There are also people who have cancer, and that is the ones we have the microgrid data on. They have cancer, so you need to map what you're going to take out. And so that's not normal cortex, but it's quasi-normal. And then there are people who are paralyzed, and that's normal cortex. So we don't know, we don't have a way to measure these in most people, and just non-invasively.